Thank you, everybody, for coming to this Linnean Society lunchtime lecture in the International Year of Plant Health. We're very lucky to have Sophien Kamoun here with us today. Sophien is an amazing person who I first met at a Solanaceae meeting many years ago. He is Tunisian, but as you may have just heard with our chat, is a dual citizen of Tunisia and the United States. He went to university in Paris and then went to do his PhD in the United States at the University of California at Davis. He worked then at Davis for a while after that, then went to Ohio State, which is when I first met Sophien, when we had a huge argument about the scientific name of the tomato over dinner one time at a plant and animal genome meeting. I'll never forget it, at which we just went into why taxonomists change the names of things. He then moved in 2007 here to England and to the Sainsbury lab, and is a world expert on plant immunity. Now, many of you who perhaps have plant awareness deficit, or what was formerly known as plant blindness, might not realize that plants have immunity as well. But today, Sophien, I'm sure, will tell us all about that. In 2018, Sophien was made both a Fellow of the Royal Society and received the Linnean Medal for his services to plant biology, and he's actually the first microbiologist who's ever won a Linnean medal, which is a really good first for us and something that we hope to continue into the future. So today he'll talk to us about keeping up with the plant killers, and anyone who's not convinced that plants have immune systems, after this, I'll be surprised. Over to you, Sophien, and thank you so much for coming.
Well, thank you so much, Sandy. It's been a pleasure, and it's been so much fun to interact with you over the years. And I also vividly remember that dinner when we first met back in San Diego. And I mean, I think the exciting thing with Sandy is, although our specialties are a little different, Sandy is primarily plant taxonomist, and I'm more of a molecular type. We just share so much in common because we just love nature and love biology, and are continuously amazed at all the diversity that's out there. I mean, I grew up basically being really in awe at diversity of biological systems, and although my career went more in the molecular route, I always kept that link with natural history, and I'll try to bring some of that today. And as a matter of fact, I was working on the talk this morning, and some of you may have seen the tweet, and I really struggled with focusing on this because of what's happening currently in the US. So I thought, hey, let me just start the talk with a map of the USA. And as you can see very clearly from this map, it's a very divided country. And I don't mean politically, I mean geologically. And you can really see here the Rocky Mountains being that great divide between East and West in the US. And if you have the chance to travel there, in happier times, we roam the world on biological discovery trips. And back in 2007, I traveled to Gothic in the middle of Colorado in the Rocky Mountains. And if you go actually through this ghost town, this is actually an ancient mining town which is deserted, so despite the sign. And if you go past it and go up the mountains, you'll end up in these really beautiful alpine meadows. This is like 3,000 meters in altitude or so, and you can stroll through these meadows, and you'll see the following organisms, and I would forgive you if you think they are flowers. Many actually students who go on collecting trips will confuse them with flowers, but the fact is these are not flowers. These are plants infected with a fungus, and this fungus is Puccinia monoica. It's a rust fungus, and when it colonizes its host plant Bucera stricta, which is shown there on the right side, the classical Brassicaceae, with you can definitely recognize the typical flowers, then it's completely reprogramming this host. It's taking over the biology and development of this host plant and turning it into what we like to call a zombie plant, because this plant is not going to flower. This is actually game over as far as the plant goes. And what you see there is essentially leaf arrangement, where the fungus has changed the development of the plant so that these erect leaves are actually turning into this, it's actually noxin deficient, we know that now, phenotype, and it's essentially mimicking a flower. And these yellow flowers, which also produce a scent, the fungus happens to actually shut down the production of the plant volatiles and makes its own, which are very attractive to insects. The fungus is now turning the plant into essentially a flower mimic to attract insects, which are essential for the fungus reproduction. So basically, the fungus is using the plant to attract insects and promote its own reproduction. But I love this case because it's a beautiful, vivid example of how pathogens can really take over and manipulate the host plants. And this is, Puccinia monoica is not particularly important in agriculture or in crop systems, but this is a beautiful, really beautiful, vivid example of what plant pathogens do. And you can imagine at the molecular level, all the modifications that are taking place when this infection happens. And we did study a bit of that with one of my students a few years back. I should mention that the rust fungi threaten our crops, and this is the primary reason why we have the International Year of Plant Health this year 2020. Many of these pathogens cause very important, very serious diseases. You can see that this is dating to April 2020. So while we were all thinking about the pandemic, another pandemic, there was also a plant disease that was really threatening our wheat crop. And that at the moment, when you have a new race like that popping up, fungicides are pretty much the only solution to manage it. And rust fungi are just amazing, but just the sheer number of spores they produce. You see this orange cloud behind the tractor. This was taken on a wheat field in Australia, or if you go on a stroll in these fields, then you'll be covered with spores. So the beauty of here is, just like viruses, just like the coronavirus we keep hearing about, you just actually can see the spores. And the sheer number, the sheer population size of these pathogens make them absolutely formidable evolving machines, if you like, that can keep on producing new races and overcoming whether it's plant resistance or fungicides. So a few words about the potato blight, because this is what I worked on for most of my career. So this is Phytophthora infestans, plant killer. And you can see here a resistant plant in the middle and susceptible plant around it. It's very clear that when the plant is susceptible, it's completely wiped out by this pathogen. Of course, it's infamous as being the Irish potato famine pathogen. Back in the 19th century, it really triggered those dramatic events that led to so much death and immigration. The little tidbit of information that always sticks in my mind is that the population of Ireland has never recovered to pre-famine levels. So that just tells you about the impact that this pathogen has. Here's a video that a former student, Remco Stam, has produced. This is a time lapse of potatoes sprayed with spores of Phytophthora infestans. You just see that it's very sad. The music is very appropriate. And you just see that in just a few days, the plants would be completely destroyed. Sorry, Sandy, I know you love your potatoes, but this is what Phytophthora does to potato. And just from a few spores, they'll be able to penetrate the plant tissue, the leaf tissue, and really colonize it very aggressively. And literally, the plants will be dead within a few days. I think this time lapse was done over 4 or 5 days maximum. If we zoom in on that leaf and use some molecular tricks, like in this case, this is a Phytophthora infestans that's fluorescent in red because we express a protein from a jellyfish actually in this Phytophthora to visualize it. And if you just put a few spores in the middle there, you'll see this network of hyphae that is growing into this plant tissue. And right at the edge, you can see that the pathogen is doing a very good job infecting very healthy, very green tissue. Somehow it's evading any detection from the plant. Somehow the plant is not able to detect that it's being invaded and fight it off. So the the question here really I want to start with, what I will do now is I will really take you through some of three major concepts that came out of our field of plant pathogen interactions, which kind of reflect a little bit my career too, because one of the first questions we were interested in is how do pathogens infect plants? And there's, again, it's worth mentioning that there's a multitude of organisms, nematodes, bacteria, fungi, or mycetes like Phytophthora, viruses, aphids, insects, even parasitic plants can infect plants. So plants are continuously by all other organisms that want to feed off them. But how do these organisms do it? And what we now know, is that these pathogens and parasites are very good at modifying the host plants. And they do so by secreting proteins, molecules we call effectors. And a nice metaphor maybe for the effectors, think about snakes and the venom they produce. So when a snake bites a mouse, it's producing a venom that is, for example, paralyzing the mouse. So that venom molecule is essentially, that toxin is very similar in concept to what a pathogen does with its effectors. It's essentially a molecule that's secreted inside the host plant to modify it in a way that's favorable for the microbe. And so that's what the concept of effectors are. These effectors have targets in the host, they modify them, they alter plant processes, and that's how they colonize the plant. So a beautiful, actually, definition of this goes back to this book of Richard Dawkins, The Extended Phenotype. Way before he got into all the other weird stuff, Richard Dawkins wrote really beautiful books on the selfish gene, and then later on the extended phenotype. And essentially what this definition taken from Dawkins' book, parasite genes that have phenotypic expression in the host, is really a beautiful way to illustrate what an effector is. Basically, an effector is the gene of that effector is sitting in the microbial genome or in the pest genome, but the protein itself is not functioning inside that microbial host in cell, or not functioning inside the microbial cell, but functioning in the plant cell. And that's really what this extended phenotype is about. So a vivid example here is taken from my colleague and partner in crime, Saskia Hogenhout. She works on phytoplasma. These are bacteria that also make zombie plants. So you can see here when the plant is infected on the top right, you can see that there's no flowering anymore. It's just like what I showed you with that fungus. The plant development is completely perturbed. And what Saskia did, she actually cracked what happens here, because she found that there was a single effector, a single gene called SUB54. And when she introduces SUB54 into a plant, then that effector will be switching off plant development in a way that will basically turn that plant into a zombie plant that will not flower but keep on producing green tissue. And you can imagine that for the bacteria, this is fantastic, because it's basically making that plant live longer, produce more green tissue. The bacteria will produce more and is even more likely to attract an insect vector that will carry it to another plant. So that's the concept of effectors. It became very central in our field. And actually, we know that all these pathogens, all these parasites now, we have evidence that they produce these effector molecules. If you like, they are the plant pathogen venom. So now let's switch to immunity. And Sandy gave you a bit of a clue there. But this is actually Saskia, and we went for a walk a few days ago, and I guess we'll be doing more of that in lockdown too. When we go for walks and look at plants, they're doing fine. Let's be honest. I mean, most of these plants are surviving just fine. You will see your occasional lesion here and there, but these plants are actually quite immune to most pathogens. And that leads me to this concept that is really central to plant pathology. It's been really dates back to the early days of plant pathology. Whenever someone identified the pathogen, they find out that most plants are actually resistant to that pathogen. Now, if you're an entomologist, entomologists tend to say that differently. They say, most insects are specialists. They can only infect few plants, but that's really the same thing. So another way to look at it is that most plants are actually able to defend against most of these pathogens. Disease is really the exception. There's very few cases where a pathogen would be able to actually do the trick, find, for example, the right lock and have the right key to basically unlock the system of that plant to basically infect it. And the reason why most plants are resistant is because they have an immune system. So this is now very widely accepted that plants, just like any other organisms really, have an immune system to fight off most of these pathogens. So how does that immune system work? Well, I told you about effectors. So effectors are actually secreted to do good things for the pathogen, right? But what happens is occasionally those effectors would actually trip the wire and activate immunity because plants have evolved receptors that can occasionally detect these effectors. And there's many different tricks there, but perhaps a metaphor there is a little bit like the metal detection at the airport. So if you're trying to get a gun through security, well, you'll be in trouble because the gun will actually trigger the metal detectors. And that's what plants have. They have a very effective immune receptors, which are essentially acting as metal detectors, and they can detect things that look like pathogens or pathogen modification, just like a gun would activate airport security. So when those immune receptors are activated, then the plant will mount an immune response, and it's actually very effective and very efficient at defending against all types of pathogens, whether they're bacteria, fungi, viruses, insects, nematodes, etc. So plant breeders have been using this for years because the products of these immune receptors are coded by what we call R genes. So R genes are basically resistance genes, disease resistance genes. They've been used in plant breeding for over a century. And these days we can also use GMO or transgenic systems to move these genes from one plant to another. This is, for example, a project from our colleagues at the Two Blades Foundation, where they took 3 different genes that function against Phytophthora, against the potato blight, introduce it in a potato, and that potato is doing quite well, as you can see here, without any spraying for any chemicals to kill the pathogen. This is quite serious because fungicide use, something we tend to ignore a little bit, and it's very close to my heart, having grown up in a developing country like Tunisia, is fungicide use is actually really, really bad, not just for the environment, but also for the farmers. So farmers really suffer from using fungicides because in developing countries, they typically cannot read the labels. They don't really know what they are doing with these chemicals. They don't wear protections. And you can see this picture from a colleague in Punjab. This is a potato blight field that is actually destroyed. It's too late to spray it. There is really no integrated pest management here. This farmer, sadly enough, lost the crop. He shouldn't be bothering spraying it. And certainly he should be wearing better protection, etc. So delivering genetically resistant plants is really important. And so this leads me to the last part of my presentation, which is really timely because just a few weeks ago, we heard about the Nobel Prize in Chemistry, which was awarded to 2 wonderful female scientists, Emmanuelle Charpentier and Jennifer Doudna, that you can see here. And what they discovered is CRISPR. CRISPR is gene editing. Essentially, it's a very easy way to do gene editing. And gene editing, essentially, if you think about the genome as a book, gene editing is like a word processor. It allows you to go in and change a few letters or even remove them. You can remove sentences from your book or modify the text. And CRISPR is really, as far as biology goes, is as much of a craze as you can get because it was actually invented. Their paper, the Charpentier-Doudna paper, dates back to 2012. And very soon after that, all of us started using CRISPR. This is actually back to August 2013, just a year after that Nobel Prize winning paper came out. And several of us showed that we can use CRISPR to modify plant genomes. So how can we use CRISPR toward plant health? Well, I told you about these effectors, right? So effectors have targets in the plant. So if you think about the metaphor of a key and a lock, essentially the target is like a lock and the pathogen effector is the key. So if you can modify the lock or even remove the lock, the key won't work, right? So essentially that's the same thing. So these host targets are actually facilitating infection. If you have a lock and your thief has a key, then that lock is actually helping the thief enter your house. So essentially what we can do with CRISPR is modify the target of these effectors. And this is something that we and others have done. This is one of the most obvious and most evident applications of CRISPR towards disease resistance. So for example, in this particular case, we went to tomato and removed 49 or so letters from the genome of this tomato. And you can see clearly here that this is the text on the top is essentially the genome of the wild type tomato, and the gap there is what we remove. So very precise modification. And what that modification does, it makes that tomato resistant to a fungus, the powdery mildew fungus. So this was a proof of concept study we did, but this concept is essentially of what we call susceptibility genes, a little bit the mirror image of the resistance gene, is applicable to many diseases these days. So just to finish, and gene editing is a bit complicated because it's different from transgenics. You know, transgenics and GMOs are about adding a gene to the plant. In this case, we're actually even removing a gene. So there's a lot of debate about how this could be regulated. Europe is more conservative than other places in the world. Even Japan and the US, for example, have been more open to this technology. So it is actually these days using CRISPR. For example, my colleague Zach Lipman at Cold Spring Harbor, he made this tomato which has 3 mutations in 3 different genes. And this bushy tomato, he's saying, is quite ideal for urban gardening and for a very specific niche. So there's a lot of creativity going on with using CRISPR to do a lot of different things, especially when it's deregulated or treated as a classical breeding, as is more or less the case now in the US. But guess what? So Shunyan Wang at the University of Maryland, he wanted to actually grow this tomato and engineer it with his students. And they did that last summer. And guess what? It just got powdery mildew. So Zach's tomato actually got infected in Maryland with powdery mildew. So the next iteration now is to add that other mutation, that MLO mutation I just showed you, into this tomato. So you can see how exciting really CRISPR is, because it's now a tool that allows us to really go and edit and just essentially design a tomato that is ideal for our needs. And if you have a problem with a particular disease, you might be able to solve it. So considering that this technology was only invented in 2012, 2013, it's already remarkable that we can do this sort of things. So I really want to finish there and just remind you that although 2020 was overtaken by the pandemic we all heard about, let's keep in mind that especially in developing countries, food and agriculture and plants are really important. So I'd like to finish with this quote that I borrowed from my colleague Cristobal Wao at the John Innes Center. Medicine can cure you one day, but plants save our lives every day. So thank you for your attention. I hope there's time for questions.
Thank you very much for an amazing seminar, Sophia. And that was a really amazing romp through a lot of information. So I have, can you hear me?
I can hear you very well. And maybe I went a little too fast. I was trying to stay on time.
It's not very clear. So one thing I wondered is, because people haven't actually put anything in the Q&A yet, so I get to ask the first question, is, so in that tomato that was from the Cold Spring Harbor thing, Zach's tomato.
Yep.
Do you think that this is just an endless kind of word processing editing, or do you think you'll get something that might be stable for a while, or is it just going to be every year before something, because that may be a problem for commercializing something?
Yeah, I think the the amazing thing with CRISPR, and really the exciting thing is obviously it's possible to breed a tomato like that using other methods and so on. But this is so easy. So you can imagine even a student somewhere in a developing country, for example, taking their local tomato varieties and introducing the same exact mutations. So what is really amazing here is just how simple it is to generate this kind of modifications. Whereas in the old days, this would require a lot of crosses and maybe some complicated genetics, and especially to maintain the original features of your local variety. It's a lot more flexible here, and I think the power really of this technology is how easy it is to do it. It actually is very robust. That's why they got the Nobel Prize in 7-8 years, because it just works so easily. So I don't know if that's what you had in mind with your question, but
yeah, no, yeah, that was kind of it. And it seems to me that it's kind of a really interesting way of thinking about stimulating local industry and local.
absolutely.
There's a couple of
Absolutely.
questions. There's a few questions in the
Yep.
in the chat now. So
Yeah,
that's the
let me
thing
look
about
at that.
always asking the first question is then you get overtaken by everybody else. So can
Yeah.
Gareth has asked, can you explain the difference between the use of CRISPR and restriction endonucleases? Yes, I've been a while since I've been in this area. Go easy on me.
Okay, so yeah, so restriction endonucleases are not programmable. So if you think about, basically CRISPRs are scissors, so they're essentially nucleases, endonucleases that cleave DNA, and restriction enzymes are exactly the same thing. The only difference is that CRISPR is programmable, so you can actually the biologists can modify CRISPR to basically cut any sequence they would like to cut. And so that's really the difference. The breakthrough really there is discovering that we have programmable endonucleases. And with CRISPR, it's actually easy to program because all you have to do is synthesize a short RNA that matches the sequence that you want to target. If I go back to this cartoon, I think, is it visible? I think it is visible now.
Yeah, it is.
But the sequences shown in red, what is called target 1 and target 2, are essentially the sequences we targeted with this CRISPR system. So what happened here is the enzyme, the CRISPR enzyme made 2 cuts, and essentially it's really like cut and delete. So we essentially made 2 cuts, removed that piece of DNA, and then the cell's repair system just glued it back together. So this is actually one of the simplest application of CRISPR, is to make deletions and make cuts like that. And we published a paper also earlier this year where we removed 53 kilobases, so 53,000 letters from the tomato genome. So even something as crazy as that can be done with CRISPR.
That's great. Thank you. So John Drury is asking, he's interested in knowing about the use of CRISPR for tackling rusts and who's doing work on that. And is that showing any promise?
Yeah, so we're not doing work ourselves about that, but I know there's a lot of interest in doing this. I think the problem with rust has been finding a host plant that is amenable to this kind of screens, and because the problem is really finding that gene is more than anything. So there's a lot of basic research that is lacking in the rust system in understanding the molecular mechanism behind how rust infects plants. As far as I know, there's not the equivalent of this particular gene, which is a powdery mildew. This is the MLO gene, which is quite famous for giving powdery mildew resistance when you mutate it, and it works in anywhere from cereal crops to tomato and peas, for example. But I'm not aware that there is a good example now for us. But it's really lack of knowledge more than anything. Clearly, there must be a susceptibility gene for all these pathogens. And so the dream really now in plant pathology would be to have one S gene for every major class of pathogen. But we don't have that for the rest yet.
Work in progress, sounds like.
Progress,
another question is about potato. And it's his question, this is Aska Zamarad, and he's asking if, he's saying that potato is highly heterozygous and editing through CRISPR-Cas is quite difficult. And is it possible to analyze the data from that? Is it possible to do that?
Yeah, it's possible. I mean, CRISPR was even used in wheat, which is, as you know, hexaploid. So you have a lot of copies there to mutate. So one of the advantages of CRISPR is you can actually pile up many of these target sites. So even with wheat, for example, if you're what is known as homologs, so the genes that are present in the different genomes are different in sequence, you can still target them with different CRISPR constructs. So that's not really the problem. I think with potato, the problem is that we are still relying on these old varieties and not necessarily on breeding new varieties. So I think the potato is indeed complicated for various reasons, but I don't think the reason why potato is tricky is because of the technological limitation of CRISPR. That wouldn't be the reason.
So it's a community thing. Okay, there's another question from Amir Ali Tongani, who asks, is it possible to engineer the way, for example, NLDR IDs shuffle their IDs to sustain immunity? That's that's one's beyond me, Sophien, but I'm sure you
Yeah.
can answer that.
That's a very technical question. Sorry, Amir, this is a broad audience, so we really shouldn't get into these details.
Just
But
say yes
like I
or no,
mentioned,
Sophien.
I focused on one aspect of CRISPR because I didn't want to get into a lot of details. But CRISPR can also be used to introduce a gene in a very specific location. So one difference compared to the classical transgenics is you're kind of with classical transgenics, you're randomly putting a gene somewhere in the genome. But with CRISPR, you could actually target it to a very specific location in the genome and or shuffle maybe an existing gene with a different gene. So that's indeed another application of CRISPR.
Okay, and related to that there's another question, and this is kind of a really broad question and is a good question for this audience, is there any concern for using CRISPR crops like the concerns that have been expressed for GMO crops? What's the future of that?
Yeah. So I think the concern, in my opinion, whether it's GMO or gene editing, is not about the technology. It's really about the product you make. You can always imagine making a product, for example, a GMO that is more allergenic or expresses an allergen or a toxin even that would be harmful. So it's not really about the technology. And the same way you could imagine with CRISPR introducing a mutation that would make the plant more toxic, for example, or produce more of a harmful compound. So I think the answer to that question is that the regulatory processes shouldn't be really focusing on the technology, but rather focus on the end product and whether that product is safe or not. And in many cases, with CRISPR in particular, the mutations that are introduced are not very dissimilar from mutations that can arise in nature. In fact, you could even use CRISPR to reproduce a mutation that is already existing in nature, but in a different genetic background. So again, the concerns, unfortunately, with this whole debate that's been going on about genetic engineering and so on is hasn't been very sort of scientifically grounded or very much grounded in rationality. So my perspective on this is to really not be concerned about the technology itself, but really focus on the end product and what that end product is.
So there's a related question, and I'm going to get to the one about Phytophthora and trees in a minute, because I'm interested in that as well. But this is a kind of generalized, and it follows on directly from the question that you've just kind of dealt with, is how do you, Paberthran has asked, how do you think reservations about the economic repercussions of GMOs and CRISPR modified plants can be best addressed and the crops be made accessible to farmers at affordable prices instead of allowing monopoly by some big companies, which kind of touches on the thing that we talked about at the very beginning.
Yeah, that's actually a really excellent question. I really like that question because indeed, I think one of the issues with GM and transgenics is that deregulation is so expensive that only few companies in the world can afford to actually take a product to the market. What I really like about CRISPR, and I think that's something I've been promoting its use in developing countries. For example, I just had a session on CRISPR with Bangladeshi colleagues, which actually lasted 3.5 hours. It just shows how excited they were about the technology. And so in my view, actually, this is a way to bypass that problem of a few multinationals dominating the technology. So it is actually easy to implement. And if indeed scientists in Bangladesh or elsewhere can make their own CRISPR mutant, their own resistant plants, that would be fantastic. That would be one way to address one of the indeed concerns about GM in the past. So for example, this particular project, this tomato, we were actually able to generate this plant in 10 months. So from the beginning, from the bioinformatic work to design these constructs, to actually getting a plant with only that mutation, that whole process took 10 months. So it is a really powerful and rather simple technology to that I like to say democratizes gene editing.
Great, that's terrific. So Prithvi says, thanks for a wonderful talk. There are a lot of Phytophthora species that can infect forest trees as well. What can be the best approach to deal with them using CRISPR? Can spraying with other microbes edited against those Phytophthora be a way? And I know you've worked on Phytophthora in all kinds of organisms.
That's a very tricky question because trees are first biologically fascinating. I'm actually very fascinated by trees myself, and I also fascinated by the immune system, which we don't understand very well. It must be quite amazing. I mean, for these plants to survive so long. But indeed, we have introduced a lot of phytophthora, probably through imports of horticulture products, I guess, and I hope we will really stop doing that. There's some great projects now looking into strengthening actually our surveillance methods. I really think it's problematic with trees because as you know, it takes a long time to actually just really introduce new varieties and replace the existing ones. So it is problematic. Ash dieback is a good example, maybe better than Phytophthora. Phytophthora really was it's actually right now there's no good solution. Maybe uh, like you mentioned, using microbes or some biotic solutions like that. But genetically, it's really hard. But with ash dieback, I think there's some really good hope right now that there are some projects that identified some resistant material, or at least partially resistant material, and maybe renewing the British stock of British ash with this material may give some hope. But I think trees are really tricky. I don't have a magic wand here to give you a simple answer.
Great, thanks. There's another question asking about the engineering of plant PRRs instead of changing pathogen targets.
That's again another technical question. I was trying to avoid getting into this sort of details, but yes, that's certainly another approach that has been used. And PRRs, what the question refers to, are another class of immune receptors that are on the cell surface of the plant, and they tend to recognize more conserved features of the pathogens, and they indeed have been used to make plants more resistant. So. Yes, I mean, I can only say that that's of course another alternative,
Another possibility.
tool in
So
the kit.
Hervé van der Heyden has asked that in the tomato example that you showed, that actually the picture of it is up on the screen now.
Yep.
Were there any downside effects following the removal of those 49 nucleotides? Was there anything that you didn't expect?
Yeah, so I always get that question. That's really an excellent question, and that's a very logical question because if you think about it, you know, if you can remove this gene, and the plant is fine, what's that gene doing in the first place? You know, why is it there, right? So I get this question all the time. It's a fantastic question. So there's two answers. The first one is we couldn't see anything, um, any hits. This plant was not did not have lower fitness or did not produce less tomatoes and things like that. So we haven't tested it under field conditions, but based on work from others, we don't expect that the MLO mutation will have a major hit. The second part of the question is the way I rephrased it is why is that gene there in the first place? So I'm going to give you a metaphor, and I usually insert another slide here, which is a naked cat. You know, have you ever seen these hairless cats? You know, there's these cat breeds that don't have hair, and yeah, I mean that's really weird. And then you could ask yourself, why are the hair genes there in the first place? Well, obviously that cat is domesticated, would not survive in the wild. And the same way that our crops are also kind of domesticated and are pampered in our fields. So there are many genes that these crops carry that are very essential for them to survive in the wild, but under our agriculture pampered conditions, just like our domestic cats, will do fine without them. So it does make sense that there are probably many genes we could remove from the plant that will still make it a robust crop, but probably that plant wouldn't do well at all in the wild. An example would be the flowering genes of potato. I guess we could probably remove all the flowering genes of potato. It would be very sad for Sandy, for all of us, but that would still be a reasonably good crop, I guess.
Yeah, I love that image, Sophien, of pampered crops.
They are pampered.
One always thinks about us domesticating crops, but actually crops in their own way have domesticated us
Of
to look
course.
after them in a really super amazing way.
Of course,
So
and we do really
there's,
take care of them very well.
we do. We look after them very, very carefully.
Mhm.
Well, thank you, Sophien, for a really, really interesting talk, and to everybody for all their questions and for being here. And please do, if you're not already fellows of the Linnean Society, become a fellow of the Society, because by being a fellow, you can nominate more microbiologists to be recipients of our Linnean medal. So it's a great society to belong to because it's hugely diverse and full of all kinds of people with all sorts of interests. And and we have lots of really good seminars. So please do join if you haven't thought of it already. So thanks so much. And thank you very much, Sophien, for giving that great talk.
Thank you, Sandy, and thank you everyone for your attention.